from Microbe TV. This is Q&A with A&V. I'm Vincent Racaniello, and joining us tonight from Chevy Chase, Maryland, Amy Rosenfeld. Hello, Amy.
Hello, Vincent. How are you doing today?
I'm well. Every day has challenges, but they can be overcome eventually, and the sun shines. Yeah, it's no problem. So hello, everyone. How is everyone tonight? We have, for moderators, we have Les tonight. I saw Tom. I saw Steph. Did I see Vanity? I didn't see Vanity yet, but although I was texting with her from the train, there she is. Hello, Vanity. And those are our moderators tonight. Be grateful for what they do. It's a thankless job, but it needs to be done to keep this a civilized conversation about viruses, which is what we want. Now, I want to give you a little teaser for what's coming later. Okay, folks? You ready? Ta-da! It's a black box. It says YouTube. What do you think? You can see how thick it is, all right? And it just has an address label. Okay, that is our 100,000 subscriber award. And I will open it in a bit later because there are only almost 100 people here. So I want to wait till a few more. Some people come in later. So that should be fun. A YouTube award.
It's going to be very exciting.
Yes, and it will end up in. By the way, it's pretty substantial. I would say it weighs a pound. It's not a plushie, that's for sure. It weighs about a pound. So before that, I want to just give Amy a little attention here. And I think some
Me?
of you may. Yeah, you're Amy, aren't you?
Yeah.
Okay.
Oh, yeah, that's my paper.
So this is Amy's paper that she published January of this year. Cross-reactive antibody responses against non-polio virus enteroviruses. There she is. And,
that's me.
Then we had our student Edmund, and then I should say Amy's student and Amy's technician Michaela, and then Nishay and Ian from the Lipkin lab, and then me. Amy, give us the executive summary. What is this about?
So it's about the fact that you can't do serosurveys and take samples from people and look at antibodies and just say that they've been infected with that specific virus because there's cross-reactivity. So antibodies that bind and neutralize polio can bind and neutralize EV68 and 71 and Rhino 1A. And because it's a polyclonal sera, you don't know if it's exactly the same antibodies or if it's different antibodies or exactly what. So it's a revolutionary thought in the field. And it also makes it question whether or not how we're going to design disease, vaccines, and other immunotherapies against these specific viruses.
Now, the reason I bring this up, and we talked about this on a TWIV, right?
Yeah, we had a whole TWIV on it where Dixon went, "This is my favorite show. We've gone through all of the pitfalls. We have 2 retired professors and this professor and that professor. And then we have UAV." And I was like, okay.
What was the
It's
episode?
Called,
what was the name?
Retro antibodies or something.
Here we go.
Yeah, EV antibodies revolutionizing our thinking. Yeah.
Amy returns to TWIV to discuss her work on the identification of cross-reactive antibody responses and the implications for our understanding of viral pathogenesis and seroprevalence studies. That's from February. Okay. Now, the reason I'm bringing this up is because of this. This is a commentary in MBio that just came out by Megan Culler Freeman from the University of Pittsburgh. And this is a commentary on Amy's work. See, in this commentary, the article by Rosenfeld et al. Is put into context. And the commentary is called Setting the Stage for the Next Great Vaccine Success Story. So, Amy, what do they write? What does she write about in this commentary?
So at the very end, so she summarizes the results and she puts it in the context of, how IPV and OPV got rid of polio, and maybe it's not going to, whether or not that also had any implications in enterocirculation and our inability to see paralysis due to other enteros. And then at the very last paragraph, she starts talking about our future directions, which is
The very last, the study
well,
also is
the whole right there. This potential cross reactivity and cross protective antibody profile may also help explain why 68 circulates in a biannual pattern. And then all the way down, she says at the end, if we in turn vaccinate against non-polio enteroviruses and prevention of AFM into the next great vaccine success story today, there is much work ahead to understand both the specific antibody response required for protection and the immunological interplay between poliovirus and non-polio enteroviruses. I would have just said the immunogenic interplay among enteroviruses, and including rhinos, because we also looked at various rhinos.
So it's very nice to have a commentary about your work, right, Amy?
Yeah, it's great. I'm glad that somebody, she's a pediatrician in Pittsburgh.
Congratulations, Amy.
Thank you very much.
And I should say that this is really the work that propelled you to your new job, right?
Yeah, for sure.
Would you agree?
Yeah, it's 4 months of work. So David can say he did 6 months of work to get a Nobel Prize. I did 4 months of work to get a job. Yeah,
I think it's more of that because you were viewed as a mature scientist, right?
Yes.
So anyway, I thought we should point that out because that's a really important achievement to have a commentary made about your work. So
yeah,
okay,
I know.
And as I said, we'll do the unboxing of the YouTube award maybe around 8:30 or so.
Yeah.
Let's do a few questions. Did you guys see the meeting today?
Oh, we got questions?
Of course, always. That's what we're here for, right?
Oh, I thought it was jibber jabber.
Well, did you see the meeting about pediatric vaccines? Amy, summarize what happened for us.
21 yeses to authorize the Moderna and Pfizer vaccines for 5 and under.
It's good stuff, right?
Yeah, it's excellent. It's fantastic. Time to vaccinate. I think they're anticipating rollout beginning June 19th, which is like Sunday.
Excellent. It's really good news. As Daniel said, in June, and it's in June, right?
Yeah.
Excellent. Here's your friend, Animal Party, Amy, our friend.
Yeah,
hello, Animal
Jen.
Party.
Jen. So exciting.
Now, Tom and his wife had COVID this week.
Oh, not so good.
If you're saying you have COVID, that means you had symptoms, right? I'm hoping that you had symptoms. Well, I'm not hoping. I'm hoping that you're getting the pedantry right, or you just didn't test positive. So I'm glad you're better, and the vaccines work. Philip said, I've learned a lot about bagels. Would you like me to teach you about grits?
Sure. We can learn about grits. Cheesy grits. What I know is that when a girl picks you up in a bar and then says to you, come home and I'll make your grits in the morning, shrimp and grits in the morning, you should go. That's what I know.
Wow.
I have a friend who was picked up in a bar. Where was he? Somewhere down south. And he called and he said, this girl wants to make me shrimp and grits. I'm going to my hotel. And I said, Andrew, you just don't understand how the South works.
You know, the best grits movie that I know of was My Cousin Vinny. And I'm sure everyone will get it. Did you ever see that movie, Amy?
I saw parts of it.
Yeah, grits.
Like where he says, you youngins. Isn't that one of the famous lines? You youngins.
Yeah, well, he says ute, and the judge wants, what's a ute? These two utes here in the front row.
Yeah.
Anyways, very good movie. I love it, not just because it's Vinny, which is what I am, but I love it. Okay. Now, what else was I going to say? Oh, so I wanted to tell everyone that this past weekend I was in Washington and I went to Amy's and fixed her lights. And you can see we moved the desk and it's in a new place. And you can see her artwork in the back there, right?
Yeah, my big Keith Haring. Yeah. Pop art. Pop art. You just need a bigger one now.
New T-shirt. Short form TWIV saves lives. Long form TWIV reduces mortality and morbidity. Okay, Dania, can you get on this T-shirt request, please? Dania is working on the cafe press angle for us, which
That's great. She's making your. Did you order T-shirts for ASV yet?
I did not. I have to do that, and I want to make it an ASV specific T-shirt. But you know, things happen. Things happen that make it. Anyway, today, folks, I recorded a video at the incubator, a tour of the incubator with David, our video guy. Should be out in a couple of weeks, so look for that. That'll be fun.
Who's editing it?
He's editing it. Oh, and I did 2 pods in D.C. this past weekend. I did a TWIM and a TWIV. And I'm hoping to drop the TWIV maybe tonight. Do I dare drop a TWIV after 10 p.m. here? Anyway, it should be good. You'll like it.
Ortega is done editing? The
Yeah,
files are so big, Vincent. I can't give them to you. The files are so big.
He said he couldn't. The files were too big to get to me, so he would edit it. So he did the audio and video. He's done. He already sent me a link to download them. So I'll put that up.
That's great. Can't wait to see what they think about your guests and if they go and go shopping with your guests.
Timely Paxlovid with underlying vaccine protection did the trick for us. Well, one would think. One never knows with such anecdotal information, right, Tom? But most likely. Nadia, and this is not Nadia, this is Amy, sorry. Any opinion on the Imperial College study that previous Omicron infection or vaccines have almost zero protection against Omicron reinfections? This is not the one we were talking about, is it, Amy, with the imprinting?
Yeah, it is. I believe it is, with the imprinting and with the healthcare workers. And okay, sure.
Well, infection is not what we're worried about. We would like to know about disease. So what's the protection against disease? Did they look at that in that study, Amy?
No, it's all in vitro neutralization assays, basically.
All right, so it's not really interesting. But it's kind of surprising that the homologous virus wouldn't induce
I think that there's problems. Mucho problems.
With the study? Okay.
Yeah, mucho problemo.
Any new data on long COVID and vaccines? The last data I know of is the VA study that we did on TWIV. Was it last Friday? I guess so, yeah. No, it was the Friday before. Last Friday was a papillomavirus paper.
Yeah.
Amy, infants get their first vaccines at 2 months, but they don't have an adaptive immune system till 6. So how can the vaccines work? Well, it starts to develop. It doesn't go on like a light switch, and all of a sudden it's on at 6 months. It develops over time, and there's some of it at 2 already. And for some viruses, you need to get them immunized early because that's their target. So it's biology is never black and white. It's always gray. What do you think, Amy? Any ideas?
For sure, it's always gray. And yes, some of it can start early.
What do you think about WHO wanting to rename? It's not actually monkeypox, but rather the isolates, which are based on countries, to remove any stigma from those countries.
Are they going to talk to the scientists?
I hope they talk to Jens Kuhn, right?
I would think that they should.
I bet they will. I don't mind them remove if it's really a stigma. That's fine. So there's DRC isolate, right? You take out DRC or whatever it is. I agree. It's fine.
I don't know. I didn't know that they were naming isolates. I thought it was just monkeypox because it doesn't really vary like the same rate as SARS-CoV-2. So I didn't realize that they were doing that.
Anyone here besides me who has not tested positive? I've never tested positive for SARS-CoV-2. I don't think Amy has.
No, I've never tested positive.
I spoke to someone today who I know well who had never tested positive. Spoke with someone last week. Yeah, there are many people who have not tested positive. It doesn't mean they
I have
weren't
friends who
infected.
have never tested positive either.
But you could miss it if you don't test. I haven't tested since I came back from Zurich, which was a couple of weeks ago, right?
Yes. Last month, right before the incubator party.
If your grits have no taste, you have COVID. That's funny.
It's not always true. Grits are, to be honest with you, I know, but to be honest with you, grits can be tricky because they can be very gummy and then they're not yummy.
It's been a while since I had grits. Elena loves your print. She met him years ago at his pop shop in the village.
Yeah, that's exactly where I bought it. It was the pop shop on Lafayette. And it was right after he had died. And then the following day I went to see his chapel at St. John the Divine, which has yet to be finished. But yeah. He was the graffiti artist of the subway. Yeah, super cool.
How old is that?
Maplethorpe.
How old is that reproduction that you have there?
I bought it, let's see, it's close to 22 years old. Bought it in the very early 90s. Yeah.
Kang wants to know, are there factors that allow some viruses to retain infectivity longer than others outside of cells? Absolutely, there are, but nobody knows what they are, right? Would you say
No, but you could say that some of them are like the stability of the particle, right?
Right.
Like if you dry out like if you have a lipid envelope and you dry out versus like polio that doesn't have a lipid envelope, right?
Yeah, so poliovirus is quite stable in the environment. So is norovirus. Now these are viruses that go through the intestinal tract, which has extremes of pH, all sorts of enzymes. So they need to be very stable, and the icosahedral shell is probably part of that, not having a membrane. Amy, we used to purify poliovirus using 0.5% soap, SDS.
Yeah, for sure.
Totally retained infectivity. We used to run gradients and then add SDS to solubilize the pellet. I remember once one of my students, she took a little bit and did a plaque assay, and all the cells lysed because...
Yeah, I was going to say, so then what did you do? You dialyzed it into DMEM to get out the SDS? Because you sure didn't want to use the SDS for anything.
SDS is not easy to dialyze. I think we just diluted it because the titer is so high, you could make a substantial dilution so that there wasn't enough detergent to kill the cells, yeah.
Well, how much is a little? You did what, 0.1% SDS?
I think it was 0.5% SDS, yeah. I'm grateful for Paxlovid. I tested positive. Yes, so Paxlovid is good for those of you that have the right indications. Remember, under the EUA, there are certain indications you need to have, although I know that many physicians are violating the EUA. But that's the law, right? Vincent will have circular eyes now. Why is that? What is it? Do you understand this?
I don't know.
Okay. Jack says, what do you think of today's statement by this person from the WHO? As a result of the precipitous worldwide decline in sequencing, we're really playing with fire here. I don't think we're playing with fire at all.
I don't think you need to sequence at
No,
all.
nope. I mean, we're not playing with fire. We used to deal with outbreaks of all sorts of viruses without knowing the genome sequences. And I would say that aside from tracing the origins of certain lineages, treatments haven't changed. The monoclonals, of course, have to be tested against isolates to make sure they still neutralize them. But if you didn't have the sequence, you would just test the isolate. And no, so no, I don't agree that we're
The
playing
truth
with fire.
is we don't sequence a ton during a flu epidemic or a flu outbreak, so I'm not worried. It's just throwing technology at a problem that where it doesn't belong.
Is remdesivir being used enough with mobile IV units, hospital in home? Is it being used enough?
Ask Daniel.
I think Daniel said that it's hard to overcome the early negativity about remdesivir, right? Everyone wants Paxlovid now. And so probably not, I would say.
Hey, is he the one who gave you the blue Eddie? You should tell him he
No,
made a
no, no.
video. Oh,
I don't think so. No,
he's
I
not the
don't.
guy from New Zealand?
I don't think so. No, I think it's
Oh, I
someone else.
thought it was.
Here's a different icon.
Ah, well, icons change, you know.
Icon Schmicon.
Yeah, pretty much. Animal Party said she wanted no longer to be Animal Party. Then I would never know who Jen was.
Well, you just use her name, right? Animal
Yeah,
Party.
but you see Animal Party, and then I know it's Jen.
What do you say to people who are pretending COVID is gone? You're wrong. And they'll say, how do you know? Because I'm a virologist. I know these things. And then they say, well, I don't really care about expertise. And then at that point, just go on with your life. It's not worth it. What do you think about that, Amy?
Sounds good.
Is that what you would do if someone came up, let's say your cousin in Boca Raton came up to you and said,
She
Amy,
wouldn't say something so stupid.
I'm doing a hypothetical, okay? What if your cousin in Boca said, COVID's gone? What would you say to them?
Okay, where'd it go? Because if it's on vacation, I want to go there.
Where did it go? That's a good response. I like that.
Yeah,
That's
where'd it
actually
go? I
excellent.
want to know. I want to know if it's
Huh.
a play like the lady that you had the call with that you said was in Bali.
Yeah.
Then I want to go to Bali.
If COVID went to Bali, yeah.
Yeah, it looked really nice there.
I often ate spicy foods. After J&J made my head itch and sweat. I got COVID and the new spicy sensitivity dwindled and is gone in a month. Link. Possibly. Yeah. Alterations of taste and smell are clearly occurring with COVID. And so, yeah, it's probably related. I'm not sure it's not related to the vaccine, but it's related to your infection. Yeah. Why does our immune system tend to diminish over time as we age? Would a
Because
major
you're not
factor?
supposed to live forever.
Yeah, all you need to do is get past reproductive age, all right? And then systems evolutionarily did not evolve to last 80, 90 years, right? Just to get you
Right.
past reproductive age. And in fact, women's immune systems at reproductive age are much better than men's. And that's to protect them. And then once both age, they're both equally poor. So it didn't evolve to be long-lived like many other aspects of human or animal biology, yeah? I don't think it's accumulation of all these things at all, no. Okay, now, I've done an EBV antibody assay. IgG was over 600. Standard range is 0 to 18. Monocytes, 8%. Could a Tdap booster reactivate it? I don't know.
I've never heard of that.
Why would a Tdap booster do anything. I don't know that. No,
Yeah, I've
it
never
doesn't
heard
make
that.
sense to me. And another question: how effective is hydrogen peroxide at inactivating viruses, bacteria, and parasites? It's really good. Yeah, it's really good at inactivating them. But we don't
So
use
should
it
we
for
all
that.
ingest it?
No. It's not like bleach. No. No, you don't want to do that. If you had COVID and test negative for antibodies, would that indicate your innate system was the only one involved in targeting the virus?
Not necessarily.
I'm sure that
You didn't test for T cells.
I
Didn't
bet your
test
T
for
cells were.
complement.
I bet your T cells complement and maybe even NK cells, right?
Yeah, exactly.
My cousin tested positive for COVID 2 weeks ago. Was symptomatic. I'll see him this weekend. He may be shedding, but does that mean he's still contagious?
I'm not sure he's shedding. And number one, and just because you shed, you may not be contagious because you probably don't have enough to transmit or initiate another infection, since we don't know anything about how much is required. Right?
Yeah. It's not given. Not a given for sure. All right, back to Ian. With vaccines that produce high antibodies, are not high antibodies correlated with later B and T cell strength?
I
So
don't think
Go ahead.
I don't think number correlates with quality or is quantitative to. I don't think that there's any direct correlation to binding and stuff.
No, because the antibody response evolves also. So I don't think it's indicative of memory in B and T cells.
Exactly.
How hard would it be to experiment on chimps? It's not
allowed anymore.
It's not allowed. It's illegal in the U.S. Can't
I think it's
experiment
illegal
on chimps.
throughout the rest of
The
I
whole
think
world?
that's yeah, I think it's a WHO thing too.
Yeah, we used to do a lot on chimp. Not we, but investigators used to, but no longer. The return. You're twisting something there, Amy. And
Sorry.
it makes a lot of noise. Sorry. And we're not allowed to do it anymore because they're too much like us. From Ireland, this podcast, Amy, all I can say is you got it, girl. And Vincent, love all your podcasts. Keep it up. Thank you, Alice O'Brien from Ireland.
Yeah, it's great.
The study you published, Amy, sounds very important for developing future vaccines, I think.
Yeah, it is.
Amy's work is seminal. It's great. And I cannot wait to see where she's going to go with it. This is the cool part. Where is Amy going next? Let's keep our eye on her. Right?
Going to bed. I don't know where I'm going. Maybe to bed.
Well, you can do that when we're done here, sure.
That's what's next. Bed is after this, therefore it's next.
Why are sharks' immune systems so robust? So I can't answer this. I don't even know if they're robust. Are they?
I don't like the word robust, so I don't know.
All right. Holly has an anti-vax relative who takes low dose of Valtrex to prevent COVID. So this is the herpes antiviral, right? Valtrex. Correct?
So why are we taking it to prevent SARS-CoV-2 infection?
Doesn't make any sense. It has no effect on SARS-CoV-2. So the dangers of this, well, Valtrex is a relatively safe drug, but the danger is that your relative is likely to have latent herpes virus, and she may be selecting for, or he may be selecting for resistant viruses. So if he ever had to be treated with Valtrex, it wouldn't work. So that's the big danger in taking this for a long time. What do you think of that, Amy?
Yeah, I agree, but I still don't know why I would take it.
No, there's no reason to take, but Holly wanted to know the dangers, so I think that you're getting all your herpes resistant, and then if you need to be treated one day, you're done, and you could die. Thank you, Kip and Laura, two Farm Ds from San Francisco. Thank you very much. Very kind of you. And yes, the award, well, we passed 100,000 some time ago, but it was someone here, maybe it was Elizabeth actually, who's, I said, I couldn't get it. And she said, here's what you have to do. She was very calm. And I did it, and here it is. I ordered it. You just order it. You don't have to pay for it. So very excited to unbox it. Should we do that now?
Yeah, when's it going to happen?
You can do it now.
It's 8:30. It's
Okay.
8:30. That's 427 peoples.
Okay, we're going to unbox it. So I actually have not opened this at all, okay?
Well, it's not centered. Nobody can see it.
Well,
Okay,
can I
put
turn
it in
you
front
off
of
for a
your
bit?
face.
Can I turn you off
Sure.
for a bit?
You can turn me off. Not a problem. I'm extraneous.
Oh, you're not extraneous. I'm not going to turn you off. No.
Yeah,
Okay.
turn me off. We want to see the big unboxing. Turn me off.
All right, you're off. I'm going to put you back on, okay? Let's see.
No,
There you
you got to unbox.
Okay, we're going to unbox. All right, chill. So here we go. And we have 2 pieces of tape here. So I have a scissor just to show you that I didn't do this, okay? This is a real unboxing. So we're going to slice that piece of tape there, and then the other one. Sorry about the mic noise. And then let's open it. Oh, I got to slice this one here. Slice, 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 always away from you. Let me handle this here. There we go. Nice black box. So there we go. Oh, and there's more? No. Here we go. Ta-da! Oh, it's just a piece of it's just a piece of foam. There's nothing in it. Oh, I have to look under it. Ta-da! YouTube. All right. I have a little card here. Congratulations on your subscriber milestone. We are honored to take part in recognizing your achievement and want your experience to be exceptional. Okay. And here's a letter from YouTube. Do you remember your first subscriber, your 100th, your 1,000th? Chances are you do. And we know that you'll definitely remember your 100,000th. Your fans may have found you while searching YouTube, learning about you through a friend, or maybe you showed up as a recommended video. No matter how they came to your channel, fans stayed and their numbers increased because of your unique voice and the excitement of being part of the growing community that you established. We're thrilled to see the development of your community and proud to honor your impressive milestone of reaching 100,000 subscribers with the Silver Creator Award. Congratulations. We know you have many more stories to share with your community, and we know that your fans can't wait for you to engage and amaze them even more with your commitment and creativity. So keep creating, keep building. We can't wait to see what you'll do next, and we're here to support you along the way. And who knows, when you reach your 1,000,000th subscriber, we may just write to you and ask, do you remember your 100th subscriber? So this is signed by Susan Wojcicki, the CEO of YouTube. There you go. It's a nice letter, and you know what? This paper is pretty heavy. You can hear it. Pretty heavy stock. Okay, and now on to the award. What is this? Oh, look at that. Whoa. Presented to Microbe TV for passing 100,000 subscribers. I couldn't have done it without Amy and all of you who passed the word on. And now we'll get rid of the box, and we'll do we'll take off the plastic. This is pretty cool. It's very shiny. Look at that. So the back is felt. It's got 2 little areas where you can hang it up. And the front looks like a YouTube television, right? Oh, look at me reflecting multiple times. Look at that. That's cool. And I love the inscription. Microbe TV is there. Woohoo! Thanks, everybody. And thank you, Amy, for being part of it. Do you like that award?
Yeah, it's
Isn't
good.
that cool?
I like it. I think it's great. It goes on the shelf at the incubator. Yeah. You
Very
can now
nice.
be Earth's Virology professor, YouTube. I don't know. YouTube star.
So here's the question, everybody. Is it even possible to hit 1 million? Because many people get 1 million subscribers. I know many of the people I follow have 1 million. But we have 110,000 now, and unless we change what we do, it's going to be very difficult to get 1 million, I must say. I'll be dead, but maybe someone else will take over my channel. Maybe Amy, can you take over my channel after I'm dead?
Yeah, I'm going to take over the whole kit and caboodle. The whole enterprise will be run by me.
Well, it's a
Like,
public company. It's a nonprofit is owned by the public, so nobody owns it. But
I didn't say I was owning it. I said it's going to be taken over by me, the whole enterprise.
anyway,
It's like
thanks
Star
everybody.
Trek. Isn't that the thing? Isn't the Star Trek ship called the Enterprise?
It is called the Enterprise. Yeah.
I don't know. I didn't watch it. I've never watched Star Trek. Now I'm going to get a text from Jeff who's going to tell his friend Bill that the girl who never watched Star Wars, who never saw The Godfather, has no idea about Star Trek either. What a disaster.
That's funny. Anyway, it's very, very fun to present that here. Folks, I unboxed it for you because you asked for it. And also because this really helped push us. When we started this live stream, we were well under 100,000. I think this really helped. So this is the place to do it. And it will be displayed at the incubator. And if you visit, you can see it and get a picture with it. Okay, let's go back to questions. That's what we're here for, right? How long, Jeff from Omaha, who I met, how long until SARS-CoV-2 turns into a common cold corona if it is in fact destined to be one? I love how that's worded. It's very perfectly worded. I don't know, Ralph Barrick first suggested this the first time he was on TWIV in the COVID era. And he said, I don't know how long this is going to take. This could be 5, 10, 25, 50 years. No idea. So that's what, and he's the expert. I'm not the expert. How long do you think it'll take, Amy, if in fact it will happen at all? Oh, look at this. Your background is all dark.
Yeah,
we can't see Keith.
No more Keith.
Do you need some background lights, Amy?
No.
No? Okay.
No, I do not need a hair light.
All right, so here's one for Amy. Based on your work, how do you think about some of the serological surveys suggesting SARS-CoV-2 was circulating in 2019?
Well, my work is on a different family of viruses, and those family of viruses are extraordinarily genetically similar, which makes the probability of cross-reactive antibodies to be way greater than there are for SARS-CoV-2 and SARS-CoV or MERS. And so when you're able to discern and you have a specific antibody for both of them or all three viruses, then your serosurveys are much more accurate than when you just have a hodgepodge, which is what my viruses are, because something went wrong.
Los Angeles is on the edge of going back to indoor mask mandates due to 6% rise in hospitalizations, even though with or due to COVID is not discussed. I think that's a very good point. That I don't think hospitalization is the good metric, and many others feel the same way. I think you need to look at deaths, and I'm not sure they're going up. And here's another mention of the paper that we already sort of addressed, the article saying Omicron infection not inducing lasting immunity. Sounds a bit off. Yes, Amy agrees that it sounds a bit off. And I think, Amy, should we do this next week on TWIV? Is it worth doing?
Sure. It could be a snippet.
Okay. So Greg says, in terms of data storage limits, how many viruses can our body be trained to remember and fight? Ooh. 1,000, a million, no limit. How many vaccines can an individual receive before storage is exhausted? Well, you have a huge capacity for recognizing antigens, but you can't keep memory cells for all of that around, right? It's just too much. So you have to limit it.
Well, you would like your blood to move places. It's kind of helpful when it moves oxygen from the lungs to the brain. Otherwise, it wouldn't be so helpful.
Exactly right. You need to have that blood moving. And so I don't know that there's a limit, but there's a limit to the number of B cells, memory B cells that can be circulating. And I'm not sure what that is. It's a good question. And here's another contribution from Kip and Laura. Nice work, Amy. Thank you so much. Really appreciate it. Okay, D614G. With a typical academic lab needing some DNA, something like insertion to make experimental adenovirus vector vaccine, cook it in-house or have it made by a specialty supplier? I think Amy has had DNA made. Have you? Or no? You always clone it yourself?
Not outside oligos. Yeah, not outside oligos.
I would say that companies have it made.
It depends on the wealth of the lab, to be honest. Totally depends on the wealth of the lab.
And to be honest, Amy, we were not a wealthy lab, right?
Us? Wealthy? Money? Oh, no.
So we used to make our own DNA. But you're right, Doug. If you have money, you would have it made. So Amy, if you wanted to synthesize poliovirus, 7,400 bases, how much would that cost? A few $1,000s?
It's close to 10.
Close to 10. If you wanted a gene, you could synthesize it. You could have it synthesized. So the way we would do it is we say, okay, who has this and give it to us, please?
Or just RT it up ourselves.
Yeah, we could reverse transcribe it up. It's a good question. Very good. All right, Jeff is here. Hello, Jeff. Watching us on the big screen, no doubt. I have a really novel suggestion. Let's all get vaccinated and move on with our lives. No more mask theater. Just accept that the vaccines do what they were designed to do. Yeah, now that we can immunize kids below 5, this is good, right?
Yeah, it's great. It's going to protect against the development of severe disease and death. It's good.
Cat Vaxxer said, gives us a nice little contribution. Finally tested positive for SARS-CoV-2. Fully vaxxed, mild infection. Vaccines work. Thank you for all your efforts during this pandemic. You're welcome, and thank you for your contribution to the incubator, which we wish to continue to make science education for everyone. And it's going to change, right? Because right now it's molded in my vision. But as other people come in, it can be changed. The vision can change somewhat. Absolutely. I have no problem with that.
Well, it has to change with technology and stuff. So, yeah.
I have COVID this week with symptoms, but both my daughters old do not have symptoms. Yes, this happens with neither rhyme nor reason. Okay, back to primer and primer. No native UK speaker pronounces primer. We say primer. For 500 years ago, we said primer. Okay. I've heard people from the UK, scientist types, say primer. Okay, that's why I said that. Maybe you never heard a scientist say it, but I've heard it.
Whatever, move on.
Do you think the response to African swine flu in China contributed to the outbreak of SARS-CoV-2? The lack of pork left a hole in the supply of protein that had to be made up by wild game. I don't think so. Wild game is way too expensive to be a substitute. I think Peter Daszak talked about that on one of his TWIVs. He said, no, it's too expensive to be a substitute. The Two Utes, right. The Two Utes, great movie. Okay, so Tom was symptomatic. Malaise was really bad. Other cold-like symptoms, major fatigue, good response to COVID. Good. I'm glad you're better. I'm very happy about that. Salutations to my favorite virologists. Could you even say you had a favorite virologist before the pandemic? Did you know any? Isn't that funny?
It is.
Les got COVID by rapid test / symptoms. Took Daniel's advice and it was mild. Very good. Oh, here's Garrett. Hello, Garrett. Garrett visited the incubator last week. We had a great time. My wife now partly understands my obsession with TWIV. Wish we had made the opening the week before. So Garrett visited. Remember, Amy, I told you he was coming. picked up his
Yeah, I
textbook.
remember. He picked up his textbook. Yeah.
And Garrett will tell you it's fun to visit the incubator because it's a cool place. It's not only has a great Ebola painting and a bunch of other good science art, but it's got a cool studio that I put together. And it's decorated by Amy.
Yeah, it's good. It is decorated by me. It's going to be, it gets 2 more chairs and then we call it a day. No more stuff.
How does sugar consumption affect a COVID infection? I have no idea. What about you, Amy?
I have no idea unless he's trying to like refer to people who are diabetic, and diabetes is not necessary. I mean, it can be triggered by sugar consumption, but that's not the only thing.
Did you hear Dr. Fauci tested positive? He had some symptoms. Is that correct, Amy?
I have no idea.
Mild
I
symptoms?
sent you the article. Yeah, maybe. I sent you the article. I'm not that concerned.
So Hotez also had COVID. Is that right? Okay.
Could be. I don't know. No, keep track. I'm not that interested.
Israeli article, BA5 causing more hospitalization. Yeah, but hospitalization is not a good metric. I'm sorry. I just think it's flawed, and I don't understand why people are using it because
It's easy.
It's easy. All this talk about grits and shrimp is making me hungry. Glad I was able to get on into Twiv. Parts of my area in Wisconsin was under a tornado warning and thought I'd miss this live stream. Oh, there's Eddie with the cute dog. Remember the icon?
Yeah, I do. Not good about the tornado. Be more concerned about the tornado than anything. The house began to twitch when the witch came on a broom and then something, I don't know. Great fiasco.
Are there any
It lands on you.
Are there any trustworthy numbers on long COVID rates?
No.
I don't think so. I think they're all flawed to some extent and for a variety of reasons that we've talked about. But the last paper we did, which profiled the VA population, many VA individuals, but it's VA, it's not the general population. And the vaccines did decrease the hazard ratio of, or the risk, say, of getting long COVID. But the problem is that when you say decrease the risk, and they're looking at people with just one of the many different signs of long COVID, and I'm not sure that that's accurate. And we're not that long out either. So how long is long, right? There's lots of issues that if you calculate it now and you say, half of people who are infected get long COVID, in a year it may be down to 10%. So I'm not sure the validity of it. Oh, I think Tedros did say they were considering renaming monkeypox because it didn't come from monkeys. So then they should rename cowpox too, because that didn't come from cows. But they're not, because it's not causing cases now, right? So this is a politically driven thing. What is it? There are many viruses that are named wrong, but they're not going to rename them because they're not interested. But WHO is interested in this one. So I don't get that, really. I think you should leave the naming to the naming committee. Don't you think, Amy?
For sure. But I also think it already has a name. So no need to have a new name. It's not hurting anyone to call it monkeypox. So just move on. Don't waste my time with this frivolous frap.
Yeah, I mean, the scientists who work on it know, right? So what's the difference?
Yeah, for sure. I mean, just when you talk to Rich about like all the different poxes and where, like, what was in Jenner's vaccine is a total disaster.
Please excuse the puerile comment. Vincent is so cute and Amy utterly hilarious. You're excused, Christina. Yes, Amy is hilarious, but I'm not cute. No. I'm an old guy.
Yeah, it's all good. It's all good.
Didn't Vincent have a positive PCR test he talked about early in the pandemic, which he chalked up as a false positive. Yeah, you're right. Robbie, you have a good memory. I was wondering if
Yeah, it
anyone...
was like at cycle 39. It was
Yeah,
totally ridiculous,
it
or
was a high
42 or
CT
something. Totally
threshold.
ridiculous.
Where are you going, Amy?
Turning off the air conditioning. It's too cold.
Well, we have to get Amy a new camera because I don't like the autofocus on hers at all. I don't have autofocus, but I can focus mine with a button. Someone's complaining that it's not good. I could reach up and do it by hand, but I don't want to do that. I think that looks pretty good. All right. Yeah, so I did have, this was a false positive because I took it a week later and it was negative. And I took an antibody test and it was negative. So I don't chalk
Well, we all
that up.
know it was a false positive because it was like at cycle 42, as I said. It's totally ridiculous.
All right, so Jeff is saying, I've never tested positive. I attended large events without a mask for over a year. I've flown without a mask since the requirement was dropped. I haven't worn a mask in 4 months. I will likely test positive at some point, just saying it isn't my mask that has prevented it. Well, not everyone's infected, as you know.
Not only that, vaccines don't really, who cares if you get infected? It's the fact that they're against disease. So it's fine.
Thank you, Westfield, for your contribution. Can you briefly explain autoantibodies? Good or bad? Okay, Amy, what is an autoantibody?
An antibody that recognizes self. So some protein that is made by you, not pneumonia or shingles or something. It's made by you. So it can recognize some protein that's made by you, and then it causes an immune response. Not good.
Yeah, antibodies are supposed to be made against foreign things that are not you, but sometimes things mess up. So they're bad. Are back-to-back shots of hepatitis, shingles, pneumonia, COVID boosters bad?
No, it's fine.
You know, in the service, they go boom, 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 one after the other. And I think that's not an issue. Yeah. I never tested positive. I had my antibodies checked before getting my first booster. None were found. I continue to mask because I have a friend with long COVID. That's fine. If you have a reason, I totally respect that. And if you don't want to, I totally respect that as well. Everyone can do as they wish, but they should get vaccinated. So I had COVID 2 weeks ago. Hadn't had it since the beginning. Not vaccinated. Everybody's time will come. I'm in Florida. It's bad right now. Every day I run into people who just got over it. Yeah. It's interesting how we're in the hot days and it's still transmitting quite well. Do you believe there have been some vaccine injuries from the new vaccines for COVID-19? Yeah, there have been some vaccine-associated cases of myocarditis. There have been some vaccine-associated cases of blood clots, and some fatal, yeah. And the risk, so you have to weigh the benefits and risks, the benefits of the vaccine versus the risks of getting COVID. And if you understand how to do that, that's great. Then you can make your own decision. But most will agree that the risks of COVID are far greater than the risks of vaccination.
So this guy, Dennis, just says, my color balance is a little strange. I need a hair light. I don't need a hair light.
We're not going to get Amy a hair. She has nowhere to hang it, Dennis. The color balance is a little off. Yeah, it is.
I'm not getting a hair light. So what do you want me to do?
Can you warm it up a bit? You know how to push the warm buttons there. Do you know which way?
That's the wrong way.
I think it's the wrong way.
I figured it out. I'm a scientist.
Yeah, I know. You can figure anything out. But I'm not happy with your focus. But it's never been like this. I don't know why. It's oh, here you go. That's a good color. Take down the brightness a bit. Like one click on each side. Amy has an independent control for each light. I think that looks good. Thank you.
Yes, Dennis I have an independent controller. And then I got super controller. See, super controller.
Yeah,
Got the
which one is the
remote that
super?
controls everything. This is the super controller. And then I got controllers on each light, right?
Yeah, but you
See?
shouldn't be touching the individual light controllers, only the
Right.
remote, right?
But the super controller has individual controls too.
Yeah,
See?
your camera's also messing
Richard.
with color balance. Yeah, so we're gonna fix it. We'll get you a new one.
Okay, fine.
We
I
need.
need a new computer. Let's not get. We're not doing anything until I resolve my computer situation. My
Uh,
computer situation is getting very
will
complex.
Spike still be the target for future vaccines?
Apparently so.
Oh, it will always be included. Whether or not they expand it to include other things is another question, like nucleocapsid protein. We did a paper on TWIV recently where they had 3 proteins in the vaccine. They had spike, they had RNA polymerase, and nucleocapsid, and that was given intranasally and had some good results. So it could be that others, but spike will always be in there, yeah, because they want to have antibodies to spike that block infection, right? Yeah. Apparently, human and baboon contact is quite common in South Africa. Might this explain Omicron's phylogenetic difference from other variants?
No.
Why not, Amy?
Seriously? How many
I,
baboons you see drinking water? But
but they're
it's
sharing.
still. I don't think that they're sharing enough. I don't think it has anything to do with it. It has to do with the natural error rate of the polymerase. Plus, many of those alterations that are in Omicron were in other variants that you could see all the way at the beginning of the pandemic. So, no.
So Jeff has a good answer for what do you tell people who say COVID is gone? You say the same thing to people who say COVID is gone that you do to people who say COVID is flu. That's quite possibly the dumbest thing I've ever heard, and walk away. So that's one approach. But
I
Amy
don't
wouldn't
think
do that.
I don't think that that's a good idea.
Amy wouldn't do that. She's actually a kind person. At least to strangers, right, Amy? Oh, you got it, didn't you? You got it. We got it. Let's see. Will there ever be a therapeutic antiviral or anything that could get rid of this pandemic for good?
No.
Can you speak to more how antivirals early on could have prevented the pandemic? I can't because Amy will get mad at me because she doesn't agree with me. I always said if we had antivirals in China at the beginning, we could have stopped the pandemic, right?
Yeah, you say that all the time.
But you don't agree, right?
I think it's fine to have had antivirals. I don't know that you could stop a pandemic that was asymptomatically transmitted with antivirals. I think it's one thing to say that if there's always symptoms that so you can identify the infected. But if you have tons of asymptomatic people, what are you going to do? Give out antivirals like vitamins? Here, have some vitamin D and take your antiviral?
That's a good point. Yeah. The asymptomatic rate is
I don't know.
problematic unless you wanted to treat everyone, which you wouldn't do. Yeah. I think it's a good
Well, that's
point.
what I just said.
Yeah. All right.
Yeah. I think it's different if you wanted to say you could control a SARS or a MERS outbreak or a pox outbreak because everybody's symptomatic.
Yeah.
Are you going to control a polio outbreak?
Vaccinating with OPV.
Well, if you didn't have vaccine, how would you control a polio outbreak,
Without a...
I mean, that's part of the problem. Yeah, that's part of the problem of 68 and all these other enteros is that 9/10 of the population doesn't even know that they're infected. They walk around until somebody gets some secondary site disease like the paralysis or the myocarditis or the neonatal sepsis.
Yeah.
Do you know how many kids get infected with 71 per year? It's like gazillions.
as far as
It's
ending this
got
pandemic,
it's got a replication number of 22 in the daycare center.
as far as ending this pandemic, PM, it will end just as 1918 ended, right? We have better tools with which to end it, and vaccines are one of them. It does make most infections milder. There are always exceptions. It's not going to be 100%, but at some point it will end. And I would
Well,
argue,
when you talk to Tauenbacher, you can discuss it.
yeah, I would like to talk to Jeff Taubenberger about that. Thoughts on a nasal spray COVID vax? So they're in development, right? I don't know if, I mean, just because it's a nasal spray doesn't mean it's going to be better. It might, but it would have to be tested. I don't see anything that is going to prevent the infections, right? We're getting infections, and that's part of the problem, that people assume that many infections are going to lead to long COVID. That's part of the narrative now. And we're always going to have many infections. We have many flu infections every season. We just don't test for them, so we don't know about them. And I think part of this, getting past this, is understanding and accepting that. It's not going to go away. It is not going to change. It could very well be that we have this level of infections always. But at some point, it's not a pandemic. It's not an emergency situation.
Well, it's not an emergency situation anymore. Fauci has come out and said that the emergency situation in the United States has ended, just like the Europeans.
Testing for U.S. arriving flights was lifted last Thursday or Friday. Yeah, it could have saved me some anxiety if they had done it 2 weeks earlier, but that's fine. We know the world
A month earlier.
Was it a month?
Mm-hmm.
Thank you, Doreen, for your contribution. Have another meeting tonight, but wearing my TWIV shirt, doing my part to counter the miasma of anti-think. That's great. I love that shirt, that phrase was made by a listener a long time ago. Why does 70% alcohol inactivate SARS-CoV-2 on surface or skin, and does it inactivate all viruses? What would polio
called
do?
Dehydration. No, doesn't deact. Doesn't.
No. Bleach will, though, right?
Bleach, yeah. But no, 70% causes dehydration, which cracks the envelope, and so any enveloped virus cannot survive dehydration.
How come you didn't wear your blue glasses when accepting your award? You mean the Richard Ernst? Because these aren't my regular glasses, right? These are just reading glasses for the screen. My regular glasses, these are progressives, right? And if you wanted me to wear these in the live stream, I'd have to tilt my head back. And I've done this before, and Amy says, Vincent, can you put your reading glasses on? And then I have to put them on, and then I can look normal. But if I wore these for the award, I wouldn't be able to see, because these are only good a few feet in front of me. Everything is blurry. Okay. That's the answer. Recent studies indicate Paxlovid may not work as well in those who are vaccinated or those under 65. Any thoughts? So I haven't seen these studies. Have you, Amy?
I don't think so.
So I'd like to take a look at them, and I hope they're not preprints, because if they were, they may have issues. I don't know if they're too small or not. I don't know what the numbers are. It doesn't make much sense to me, right? I always feel that if you see a result, you have to first think if it makes sense, and then look at the data and see if it supports that, because often it doesn't make sense, and it turns out that it's wrong.
Well, this doesn't make any sense because it's not targeting something in the host. It's targeting something in the virus, and the virus doesn't know if you're 65 or 4 or fat or skinny or diabetic or not. So it doesn't make any sense. So if it had something to do with like cell metabolism or something, or some kind of immune response or something or other, it would be logical. I would agree. But this is just crap, because you're targeting the virus, and the virus does not change whether or not it infects a 65-year-old or a vaccinated person. polymerase, the protease is the same. So move on.
Thank you, Sophia, for your contribution. Does data support a second booster?
No.
No, it doesn't. And Paul Offit will come back on TWIV on July 8th to talk about this, because I think he's very compelling.
Isn't he talking also about the crap about the fifth booster in the fall or something?
Oh, changing the vaccine in the fall to like the bivalent vaccine that includes Omicron or something like that. He might, yeah.
I don't know.
Yeah.
Maybe he'll talk about the childhood vaccines instead. I'll be more positive.
Sandra is an ICU nurse, mom of 2 toddlers. Listen every time, but was able to listen live tonight. Thanks for doing this every week and keeping us informed and educated. Thank you, Sandra. And thank you for your work in the ICU. I know it's not easy. Thanks.
Yeah, it's great.
Thoughts on Fauci testing positive after supposedly been fully vaccinated and boosted. This is going to happen.
I don't like the supposed. He's fully vaccinated and boosted. And vaccines do not protect against infection. So this is fine. I don't like the nastiness of supposedly. It's got a bad connotation.
Yeah.
It implies that there's something wrong with the vaccine or that he lied, and there's nothing wrong with the vaccine. Vaccines protect against the development of severe disease, not infection.
That's right. Absolutely right. And they're working since he had a mild disease. Sean says, my sister lost her taste for a couple of months. Now she's regained it. But the things she used to like don't taste the same. Yeah, I have heard this from many people. Now they don't like things they used to like. And that's one of the features of, so when the neurons are interrupted in some way by the infection, the inflammation. And when they what's the word, Amy, when the neurons regenerate? They do something to establish the synapses. And they
regenerate.
So they don't make the connections right. And you taste.
Well, they make different connections.
Right.
They prune differently, and the synapses are not always in the same spot, because in order to have a synapse and to grow, you have to make sure that the myelin sheath is in the right spot.
Okay. Now we have lots of... I should redact my address. Oh, it's fine. Everyone knows my address here. It's okay. I'm not going to redact it. Thank you for the concern, though. I appreciate it. We didn't have a scissors accident. No. These are good scissors. I wouldn't cut myself.
You use scissors and not a box cutter?
I thought of getting a box cutter.
What kind of tool guy are you?
Yeah. Mark, hello, Mark. Good to see you. Isn't it supposed to be turtles all the way down? No, turtles all the way down. Questella, so she was here early in this stream, I remember, and she said, you will reach 100,000, don't worry, and we did. Thank you for the confidence, Questella.
That's great.
Is the word scissors singular? A pair of scissors, they call this, right? A pair of scissors. 2 scissors. I guess each blade is a scissor. This is out of my league. I don't know. Heavy stock. That's why I love this group. Yeah, the paper. You know, most paper these days is garbage, thin stuff, but they're serious about their paper. Well,
it's great.
And the writing of the letter was good. Someone put thought into it. And I would say it's not a PR person. I'm sorry if there's any PR people here. I don't mean to insult you, but you can write things in a certain way. Lots of congrats here. Appreciate it. Did the COVID pandemic cause the virology lecture views to jump? Big time. Yes. But now everything is going down, which is fine. As Amy would say, it's fine. It'll be fine. Right, Amy?
Hey, that was my contribution to the conversation.
It's okay. Contributions are going down. The engagement is going down. I understand that many people needed some information. They got it, and now they're moving on. But I hope enough remain who will continue to learn virology, because we have all kinds of virology in store for you. And, of course, all the other things on our other podcasts as well. I actually didn't know what the award would look like. Yes, I'd never seen one. Where will I post it? In the incubator. It'll be on the shelf. Amy, should I put it on the shelf behind me?
Okay.
Like next to the phage pillow? We can test it out, yeah.
We have to test it out.
Yeah. Yeah, that's viral influencer. Yeah, I got it. Yeah, it appears to be a logarithmic award. There's nothing between 100,000 and 1 million. That's a lot. The thing is, though, once you hit your first million, then you really accelerate. And, I'm not there yet. Can we not talk about your mortality? Well, I only did very briefly. But I always do. That's my thing, because you should accept it and be able to talk about it. It's not a negative thing, in my opinion.
You've been doing it since I've known you 400 years ago. So that's just
Les
the way it works.
says Susan Wojcicki, YouTube CEO, rented Google her garage as a startup.
That's cool.
I don't know about all scientists.
Okay, so here's a person who's offensive. JC in the Woods. Amy just lied again on the stream. They're neither safe nor effective.
Hey, don't say it.
Know it.
It's been deleted. Don't say it. Don't worry about it, okay? It's been deleted. Okay? Chill.
Crazy people.
I have to be more controversial, and video titles have to be eye-catching. No, I'm not going to make fake video titles. I know some people do that and they get clicks, and it's not what the video says. Not doing it. Can getting a vaccine trigger reactivation of herpes shingles? Yeah.
Yeah, that's what we see.
Yeah, because you get inflammation, and that can be a stress that will trigger reactivation. Did you know you're from the future?
What future?
The future.
Oh, well, we need to talk, because this is not good.
I think it's good to be from the future.
I think it is good, but I want better technology. I want more money, and I want better answers. So if I'm from the future, why am I stuck here? I want to be futurized.
Francois said, Montreal Public Health is now recommending vaccination of all men who have sex with men with modified vaccinia Ankara for monkeypox. Do you think it's worth the half hour for a not all that active gay man?
So this implies that it's being transmitted in the same fashion as HIV, and it's a gay man's disease.
It doesn't imply penetrative sex. It just means very close contact, right? That's what you...
Right, but then why not just give it to everybody then who has close contact? Grandparents who hug their kids, or moms and dads who sleep in the same bed, or something or other. So...
Well, I think
why not just give it to everyone then?
The transmission,
because you always have close contact.
The transmission has mainly been in MSM, right? So I don't think it should be given to everyone. So if
I don't know.
So I don't know the correlation between how active you are and what the risk is in this case. I don't think we know enough about it. I would not do it yet. I would wait to see how it develops.
How many cases have there been in total? 5,000?
I don't think it's up to 5,000 yet.
Okay.
Let's look. How many cases? Someone's going to answer it before. This is from May. This is not good. This is too old. Let's look at ProMed Mail. Someone's going to tell us soon. Okay, monkeypox update. Well, the last we did on Twiv, it was just over 1,000. Okay. Anyway, so Go ahead, Amy. Make your point. 1,300 cases so far. Okay, there you go. As of June 12th, 1,300 cases. Go ahead, Amy.
I would just think that if it were really going through a population and stuff, I mean, the bathhouses are rolling now, so 1,300 is nothing compared to what goes through the bathhouses.
I think it's.
I wonder how it stacks up with the average rate of chlamydia or syphilis or gonorrhea transmission. Probably like nowhere's not even on the map. And are we concerned about giving everybody developing a gonorrhea or chlamydia vaccine?
No, we're not.
Okay.
I get a kick out of patients coming into my pharmacy after visiting the doctor and plan to go grocery shopping who don't trust experts, even they were protected in all those activities by the FDA. Yes, it's kind of ironic, isn't it? Yep. 468 watching, actually 528, only 288 likes. Please hit the like button. It helps other people discover us. And some of you forget to do it. It's fine. And now we're reminding you. No problem.
You can push until your finger falls off.
Well, no, you can only do it once. Because if you do it twice, it turns it off. It's like an on-off switch.
Oh.
Look at this one. Our epidemiologist on TV was asked, when will the pandemic end? She said, when cases completely disappear. So according to her, the 1919 pandemic is still on. TV gets the worst experts. That's right.
That's true.
It is true. I don't get it. But you all know that.
But the truth of the matter is she doesn't know. She doesn't know any history, and she doesn't know what she's talking about. It's just a shame.
Think her internet is broken, Amy?
I don't think it was ever connected.
Mayo Clinic has recently suggested I have Behçet's disease, autoimmune vasculitis. Could be from a genetic component and or post-viral infection scenario. So,
autoimmune has to be a genetic component in inborn autoimmune diseases.
So there's no way that an infection could trigger an autoimmune disease?
It might bring it to the forefront, but the underlying thing is the genetic component, which is Casanova's inborn errors.
Yeah, so at some point we would be able to figure out what the inborn error is, correct? If someone wants to study
if there's enough people, or it goes through the family, just like the TLR alterations did for the herpes encephalitis that he first did. You know, it was within a family tree.
Yeah. So my 7.5-month pregnant daughter-in-law tested positive for the second time since being pregnant. She went in for monoclonals. Are these even working? Yes. So there are two that are working, right? Bebtelovimab and Evusheld. They both, but you won't get Evusheld for therapy. That's only for a preventative. But Bebtelovimab still works.
Changed. I think it's changed.
To what?
That it can be used as therapy. You can ask Daniel, but I think it's been changed.
No, so they did a trial and it looked good. And I asked him on the last update, what do they need for an EUA? And he said, well, they have to apply for an EUA to be able to use it as therapy. You cannot just start using it based on this trial. That's what he said.
I thought that they were applying.
Thank you, James, for your contribution. Really, really appreciate your support and your idea that you're contributing to a good cause. And we can't assume that always.
I like that. Is that the what was it? That's not the Cheshire Cat, is it? His little icon. It looks like he's like with the hashish pipe.
Well, that's a dragon, isn't it? I don't know what it is. Actually, there was
I don't know.
Here, I skipped this, but I meant to do this because he said he or she says, would the SARS-CoV-2 bats have ACE2 receptors or just similar? Similar. They're not the same. They're not the same, right?
No.
Have any American bats been found to harbor any concerning viruses that you're aware of?
Heartland.
Heartland
Or
is a
is
tick.
that rodents? Oh, it's a tick.
Yeah, I'm not sure that we've done enough sampling, but I'm not aware of any concerning viruses. But as Amy would say, how do you know what's concerning, right? What is concerning?
Exactly. I think you should talk to Tony Schouts and Peter Daszak at the BAT meeting.
We are. We're going to the BAT meeting this summer. We're going to do a TWIV from there. Yeah, that's a good question, and we'll save it for them. What do you think of that?
That was what I said.
Amy, is polio reclassified as BSL-3?
In some places, yes. Actually, if you are a new polio lab, I believe it's a BSL-3. I believe we were grandfathered into BSL-2. And I think at the FDA, it's a BSL-3.
So that's
because
just,
of GAP 2.
Is that just types 2 and 3, or is it all 3?
All 3.
It's BSL-3 now.
I believe that when I go next week, if I want to do any of it, I have to get my little gowns and my little respirator thingy and not be so happy.
Mm-hmm.
Not going to be so happy.
Thank you, Old School, for your support. Is there a best treatment for long COVID? No, Daniel has said there's nothing you can do. No, there's no best treatment. You may have therapies that you try, but there's no best treatment. No. We are going to have another live event in the fall. Okay. In the incubator,
Yeah,
Jeff?
we are.
Oh, yes, at the incubator.
What do you mean? Why are you asking him?
Who?
Why are you asking him? I'm the
Relax,
social
Amy.
coordinator here.
Relax. Jeff
And yes, we are.
Relax. Did you get your egg chair yet?
No.
Okay. Any news on avian virus? Lots of dead birds in Australia. Anecdote. It's an anecdote. I haven't heard anything about this. Have you, Amy?
No.
I love Amy's dry humor and authentic geeky personality and intelligence. What do you think?
I guess I'm a geek. I didn't think I was a geek.
Geek is a good thing.
Okay, I'm a geek.
And so this is an interesting question. Don't viruses feed on sugars? So they don't, but they need energy that is produced by metabolizing sugars, right? Like rhinovirus, right?
Yeah.
I don't know why you're so out of focus. Can you move back a little? There you go. There you go. Now it's snapped in. Maybe you were a little too close. Don't lean on the table, okay?
Okay.
Sorry, Amy.
Don't do this. Don't do that.
I'm sorry.
Sorry. It's okay.
After
fine.
mentioning longevity of females compared to men, would you think qualities of progesterone has anything to do with it? So we asked this of Sabra Klein, who studies differences in infections in sex. And it's certainly hormonal to a certain extent, but it's not the only variable. It's really not clear what it is, Mike. It's a good question. Hospitalization could denote more severe COVID. Hard to know, yes, because you could go to a hospital for COVID or with it, and that's the problem. And the bar for hospitalization could vary in different parts of the country, different parts of the world, so it's not really a good metric, and many experts agree it's not a good metric. But as Amy says, it's easy, right?
For sure.
Yeah.
You drive up to the hospital, you get counted. What's the problem? Not that hard. It's hard to miss a person.
My brother had a baby boy last night. Should I get a test before I go see him? So, presumably the mother was vaccinated, and so the baby should be protected with antibodies from the mother. I would wear a mask just to be careful. I don't think you need a test.
No test.
And Elise says
no testing.
The correct answer. The correct statement is we need a reliable definition of long COVID and reliable metrics in the clinic. Absolutely. I totally agree with that, and that's why it's very imprecise at this point.
It's always going to be imprecise.
Hi, Neva. How are you doing? Is it hard, very hard, costly, impossible to test for T or B cell reactivity to a virus? No, it's not. It's not impossible. It is a little bit more. It's more difficult than doing antibodies, which is why antibodies get done very readily. It can be done. It's more involved, yes. And it's not a test that a clinical laboratory would routinely do, where they could do neutralizing antibodies, right? Mm-hmm. Monoclonals, yes. Bebtelovimab is good for treatment, and Evusheld is good for therapy, for prevention. So Overjoyed says, giving up sugar made a big difference in her recovery for long COVID. Well, that's great, but you also don't know if something else you did may have been the key. I just don't know. And here's a good question. Should chickenpox be renamed, Amy?
To what?
Well,
why am I going to rename it? It's got 3 names already. It's got 3 names already. My God, it doesn't need a 4th.
Why is it called chickenpox to begin with? So the first Wikipedia says it's derived from chickpeas, based on resemblance of the vesicles to chickpeas.
That's fine. But, okay, so it's called chickenpox, it's VZV, and then it's shingles. It doesn't need more names.
Yeah, it doesn't. So I think Philip is saying that we shouldn't be renaming viruses left and right. Yeah, I agree with that. No, call it primer pox. Is it primer or primer, Visto? That's great. Mexico came to its senses, and they will finally vaccinate those 5 to 11 with Pfizer as soon as they get hold of the pediatric dose. No more plans to use the Cuban vaccine. That's great news. Good. I'm glad to hear that. As soon as they get a hold of the dose. I hope that doesn't take too long. The term monkey gets used as a racial slur, which is why some people want to rename monkeypox, especially as it comes from Africa. But yes, it's silly. I didn't know that. Okay, thank you for telling us. And Dr. Fauci tested positive, has mild symptoms, and is on Paxlovid. He's 81. So is that one of the risk factors?
Old age?
Yeah.
Old age, for sure.
Okay. What's the age cutoff? 65 and up?
I think 65 and up.
Okay. I was thinking, Amy looked especially cute tonight.
Oh, that's good.
Mortalities in LA County have stayed in single digits for quite a while, despite increases in cases and hospitalizations. Well, I think that's what you should be looking at, the mortality, not the hospitalization. So thank you for that information. It's great. I'm not blurry. I look really sharp here.
No need to rub it in.
Elon Musk Neuralink tech seems like a superhighway for pathogens to the brain. Would you worry about the sterility of this kind of brain blood barrier breach? Yes, I wouldn't want to have that kind of Neuralink. No. But there are people who have implants, right, for hearing, and they go through the skull. Is that correct? I don't know much about that. I could be wrong. Do you know about that?
I'm not sure how that. Yeah, I do know about that. Yeah, I don't believe so. I think it somehow regulates activation of the audio nerve, but I'm not sure.
What's the difference between, this is a joke, between bird flu and swine flu? One requires tweetment and the other requires ointment. That's fabulous. Don't you love it, Amy?
That's cute.
If you're upgrading Amy's camera, please consider streaming Microbe TV in 1080. So I don't stream this in 1080, I'm pretty sure. I'm pretty sure I'm doing a 720 stream. Yeah, I do a 720. So Amy has a 1080 camera, so do I. And I always thought for steam stability, I should do 720. But if you'd like to try 1080 next week, we can try it. And maybe that would help some of her fuzziness. But you see, mine is sharp as a tack, Amy. Thank you for the suggestion.
What can I say? I'm a
Kidding.
difficult child. I know.
Has anyone done the calculation based on numbers of infections overall as to statistically how often a single individual will get reinfected? I'm not aware of that. Are you?
No.
Okay.
It would be too hard because you'd have to have a different number for people who live in urban areas versus people who live in rural areas, because it's all dependent on your ability to engage someone else. I think it would be hard.
Mm-hmm. I bet
Which is the problem I have about reproductive numbers, right?
Yes. I bet the first few days of traveling to a new continent are at least as challenging to the immune system as getting a couple of vaccines close together. Yeah, I'm sure you get a whole new set of microbes, right, that you haven't seen before. Most of us are fine. Do you think it's healthy to get reinfected even if vaccinated? What will be the long-term consequences? I wouldn't say it's healthy, but I don't know that you can avoid it because the vaccines don't prevent reinfection. So that's what we have. That's how the vaccines work, and it's just not going to improve. Now, there is a certain an amount of long COVID after, even in vaccinated people, but I don't know how much it is. I think we don't know the exact number, so that could be a detriment. I just don't know.
I believe it's significantly less.
It is significantly less. What do you think the probability will be that CDC calls for a third round of booster shots? I think it's quite high. They're giving signals that it's going to be high because they've already... So for me to be fully vaccinated over... I forgot the age, 55? I forgot.
50.
I have to have
50.
2 boosters. 50? I have to have 2 boosters. I'm no longer fully vaccinated. I think that's not correct.
Well, the thing is that they say so they have fully vaccinated and then vaccinated up and then up to date vaccinated. So they have 2 different terms with 2 different criteria, which makes no sense whatsoever. So like Daniel talked about this on like a TWIV 3 weeks ago. It's like totally ludicrous. Just use one word. Make one definition. Call it a day.
Lori says, I travel internationally a lot and therefore want to be fully vaccinated. How long should I wait after my recent SARS-CoV-2 infection to get the 4th shot, even though I don't need it? What does Daniel say?
Go as soon as you feel better or feel up to it. You don't need to wait. But, whatever.
What about vaccines for children under 5 now approved by the FDA? The choice between the tiny risks of the vaccine and the low percentage of severe disease in this age. Okay, so here's the story, T. Over 1,000 kids have died, young kids have died of COVID in this country. If it's your kid, you're going to be devastated. So don't take the chance, even if it's low. Vaccinate. I wouldn't. If I had an under 5-year-old, I'd vaccinate them all. Do we know it originated in China for sure? The epicenter was the Huanan seafood market. That's the epicenter of the outbreak. That's where it began. Yeah. Wow.
But the virus could have been imported.
Could have been imported from somewhere else, but we don't know. Because as Amy would say, there are very similar viruses from bats in other countries, right?
Yeah, it's a pathway. The pathway that goes from China to the sea is through those countries, through that area. So the person could have been trafficking on the boat his product, either to or from Wuhan, right? Depending on what his product is, got infected and then moved back to Wuhan, right?
Yeah.
I don't know why this is like silliness at this point.
Well, people are curious. That's fine, Amy. I don't understand why people think not getting COVID makes them a god. I never said I was a god. People asked who didn't have COVID. I don't think I'm a god for not getting it. For any infectious disease, there are people that don't get it for genetic reasons, most likely. That's all there is to it.
Or they just have asymptomatic infections. Like for 100 years, my family thought my cousin didn't have chickenpox. Turns out he had chickenpox 100 years ago, like when he was 2, and he got no pox because as you age, you get the pox, right? And so he got like more of an asymptomatic infection. His mother never knew. So I have no problem with, yeah. I mean, I don't understand this man.
From the notes today from Moderna and Pfizer, looks like pound for pound, the Moderna pediatric dose looks better. Thoughts? I haven't seen the numbers. Probably Daniel will go over them tomorrow. And so why don't you listen to Daniel's update on Saturday morning, because I'm sure I'll ask him if he thinks that the difference is substantial enough. And here's Richard. How Richard likes your lighting.
Great.
Thank you for your contribution, Richard. With respect to the 2019 circulation, Ian Lipkin mentioned on the March 2020 TWIV that his friends in China told him about something circulating in China in December. So this leads to the realm of possible circulation in 2019. I think there's no doubt that it was circulating in 2019 and possibly even in November of 2019. That's quite clear. Right, Amy?
Yeah. The thing is you tell people, like in Ian's line of work, he gets told on a daily basis of some infectious disease that, there some place in the world is seeing. And whether he has no idea if he gets 100 phone calls, if 5 of them are going to be pandemic or if one is going to be a pandemic or 0, right? So he had no idea. It's not fair to penalize him with 2020 hindsight.
Does testing wastewater precede an outbreak? Then random
correlates with it.
It correlates with it. So as the outbreak is beginning, you see it in the water, right? You don't see it before, correct?
No.
And random testing surveillance, then administer antivirals. No, you have to only give antivirals to sick people, unless they have a risk factor, and then you give them Evusheld, right? But typically you don't want to give antivirals to people unless they're sick and positive. The Canadian government won't let people who are not fully vaccinated, whatever that means, from flying trains in our own country. What information might convince the government to dismiss this nationwide mandate? Well, so the problem is that they have their advisors, right, who they have engaged, and they are not going to ignore their advice. So I don't think you could tell them anything.
No, as my friend in Montreal would say, the government has its own smart people.
But I think they are under the false impression that the vaccine should be preventing infection. And I think that's the number one misconception at every level. And if you could get beyond that, it might help, right?
Yeah. I mean, but it's not just this vaccine, it's every vaccine.
Yes, every vaccine. How might one expect their immune response to evolve if exposed to the virus on a daily basis, as in continuous exposure? Second and third infections seem unnoticeable.
Yeah, that would be true.
Yeah, I think we're exposed to many viruses continuously, and we don't have any pathogenesis unless you're immunosuppressed. So I don't know how long that takes in a population, but
depends on the virus, because not all immunity is equal.
Yeah. Fox in the snow. I was paranoid of vaccines since the Wakefield study. Then came SARS-CoV-2. My year of listening to TWIV. I got all my COVID shots and my first flu shot. Thanks for educating weirdos like me. Well, I don't think you're
not weird.
I think you're smart to listen and make your decision, right? Many people don't want to even listen to us. They just shut us off and go on with their position. So I admire that, and thank you for your support. Really appreciate it. My company does some wastewater testing. It seemed to be a fair predictor of a surge when cases were high. I'm not sure it means much while cases are low, especially now it's in wildlife. And they think that is a good conclusion, Elizabeth. That's really good. And T, we talked about the vaccines approved for 6 months to 5 years. We really like it. And listen to Daniel tomorrow. He'll talk about it more. Yeah.
Saturday.
Did you see the article from Nature today on long-term infections may lead rarely to new variants? You probably knew, but the article is good. Did you see this one? I didn't see this today. I was looking at yesterday's articles still that Amy sent me yesterday, but it sounds good. I'm going to look at that. Yeah.
Yeah, no, I didn't see it today. I went to learn about benefits and went to the non-mandatory mandatory meetings. I wish they would just say it's mandatory, because non-mandatory mandatory is ridiculous.
Amy, why aren't any higher non-human primates a reservoir for poliovirus?
Lots of reasons. One has to do with receptors. One has to do with internal blocks. Lots of reasons.
But they aren't. That's very interesting. Humans are the only host for poliovirus.
The only natural host, because you can infect a chimpanzee via oral fecal transmission in the lab. So they're the only natural host.
Okay. What are some epidemiologists who think do a good job and a poor job? Who is the epidemiologist from, is it Brown?
Yeah, the guy who's now in charge of the COVID task force for Biden.
Yeah, what is his name? Ashish Jha, Canadian epidemiologist. Brown, yeah. I think he's very good. He said many things that I fully agree with. I think Shaman is not bad also at Columbia.
Yeah, I think the woman who's been publicized from Texas who moved to the CDC is not so good.
Yeah. I don't want to say who's bad in particular, but we like those three. So that we don't know that the pandemic began in China. And it's not great to suggest they should be on prophylactic antivirals. Well, I didn't suggest either, Laura. So go back and re-listen. We do know that it began in China. The epicenter, listen to the TWIV with Michael Warraby. The epicenter was the Huanan seafood market. There's no question about that. And I don't advocate prophylactic antivirals, no. I advocated treating symptomatic people early on, but as Amy said, there's so many asymptomatic people that it wouldn't make a difference, and she's absolutely right, okay?
Yeah, I just didn't understand that point. I'm not arguing that we shouldn't stockpile antivirals, but I don't know how I think it's one thing when you say it would be good for things like SARS and pox. It's another thing when you start talking about, polio in 68.
Pfizer says they are not asking for Paxlovid for low-risk folks since the clinical didn't show a benefit. Yeah. Well, remember, they had several trials. One was in hospital. It was to look at impact on hospitalization in high-risk patients. And then, that's correct, the low-risk didn't show a benefit. Yeah. Yet, as you know, it's being given to people who are not high-risk anyway. To reach 1 million subscribers, you will need a cat walking around in the background. That's right. Yep.
So you get most people watch TWIV to look at Alan's cat in the chair?
You're just joking. This is the man who is into fungi.
I know who he is. He's got his little mushroom.
Yeah. Yeah, as Elizabeth said, a lot of the TWIV hosts have cats. I don't have a cat, and I won't get one just to reach 1 million subscribers. So, thank you, Sophia.
Don't look at me.
Thank you for your contribution. My husband has had long COVID. He's tried many things. Tried stem cell therapy. Well, let us know if it helps, Sophia. I'm sorry to hear that, and keep it up. All right. I know it's not easy. I just admire the devotion you both have to your careers and your work. Thank you. Appreciate it. I don't even think of it as work. Do you think of it as work, Amy?
No.
I don't either, but thank you. Peter says the mods are our immune system. They are. Who wants to be innate immunity? Who wants to be B cells and T cells? I would like the moderators to say. So we have Steph, Tom, Les tonight, and Vanity. So who wants to be innate immunity? Who wants to be B cells? Who wants to be T cells, NK cells, and macrophages? I'd like to hear, or any part of the immune system. So you guys can answer, and then we'll get to it. It is closeness, but most of the cases are in men having sex with men. I agree that it should not be. So I mentioned on TWIV last week, which you may not have heard. I don't want to say this is only men having sex with men, because that's what we said for HIV/AIDS, and we were wrong. But so far, all the cases are because you have to be very close. It got into that community. That's the thing. It gets into a community and it transmits in that community. It could very well go into a heterosexual community and transmit that way as well. Okay, so it does not require penetrative sex. It just requires closeness, right? Did we ever say a disease is assisted with men who have sex with women? Well, we have a different name for it. So, Caroline, that name for that has existed for many years. Men who have sex with men is a relatively recent addition to our lexicon, right? So that's why it's different. Will scientists ever be free from big publishing companies? No. Nope, unfortunately. Here's our friend Rach from the Isle of Man. 35,000 more people here. Everyone seems to be infected worse than before or off work, which is a big issue. Should they work if okay? What do you think, Amy? If someone is positive but they are not symptomatic, should they go to work?
I think it's fine.
I think if everyone's vaccinated, it should be fine. It's okay. I know you didn't mean to be negative. It's reality. It makes me sad to think of a post-A&V world. Well, you got a long time, okay? Don't worry about it. Readings from the editorial staff of Journal of Clinical Ambivalence. Of the many destructive effects of media misinformation, the idea that vaccines prevent infection was one of the worst. Totally. Right, Amy?
Absolutely.
It persists to this day. Oh, I have to show you this. I have this letter here. Look. Where is it? Oh. Hold on. I'll be right back. Keep them amused, Amy.
What do you want me to do?
Sing.
Juggle? No singing. I could juggle. We can move on to the next question if I knew how to work this thing. Jeez Louise. If I could only work this thing.
You probably didn't get far. Okay, so
no,
this is great.
I don't know how to work this thing.
This is great. I love it. So this is a letter from Miley. Okay? She sent me this in September. Look at this. This is great. I love it. And this is relevant to the fellow I just put here, Alan. Look at this. This is great.
You have to move it over.
This is the Lexington County Chronicle, okay? COVID vaccines failing. And she put a little sticky on it. She wrote, I felt like the TWIV parrot when I wrote the rebuttal letter to the editor. Thank you for good information. How cool is that? I'm keeping this.
Excellent. Yeah.
And thank you for sending it. Thank you for, I mean, if you have similar headlines from your paper, send them to me. I want to collate them and use them in my teaching to show how.
What are you going to make are you going to scrapbooker now? Are you going to become a scrapbooker?
A scrapbooker?
Yeah.
What do the...
All right, it's 9:50.
We're going to finish at 10. Don't worry.
Okay.
I'm really happy that there are 550 people here with great questions.
I think
Aren't
it's
you?
great. No, I think it's horrible. I'm never doing this again. Find somebody else. Hey, hey, hey. Moving on to question.
What do
You're
these
blowing time.
What do these epidemiologists expect us to do? Do they want us to use mitigation forever? I'm confused. Oh, yeah, they do. They want you to use mitigation forever. That's their job. You know what China's doing? Incredible lockdowns. I sent Amy an article today that
Yes.
it's draconian. They force their way into your home. They kill your pets. They put you in quarantine. They separate your families. Unbelievable. Unbelievable. With no regard. Whenever there are a few cases, they do it over and over with no regard for individual people. And so that's their world.
Not good.
Oh, boy. Oh,
You
look at
always
this.
you always have to have an exit strategy.
Happy birthday to Thomas Weller, who in 1941 successfully cultured the virus that causes polio. Yeah, Weller, Robbins, and Enders,
And
right?
enders.
I think it was later than that. No, it wasn't 41. It was 51, wasn't it? Or 50? 49?
No, I think 41 is about right. They were using kidneys.
Just tuned in from Sydney. Love your videos. Can I ask, can you have Paxlovid more than once if you get COVID more than once? I don't see why
Yes.
not. If you fit the EUA, yes, you could. Rabies is a virus of concern in bats. Okay, so we earlier
We did that.
I'm not sure we said rabies virus, but I was
Yeah,
thinking
we
of
did that paper that they were using herpes as a vaccine in the vampire bats
I
against
mean
rabies.
I'm
Remember?
I'm thinking but someone asked earlier, are there any viruses of concern in American bats? And I was thinking viruses that are just unknown that we discover through screening, and we know rabies, but yeah, you're right, rabies could be concern as well, but we do have a vaccine, so that's good. Reading now that COVID infections can lead to clots months later. Considering that long COVID is possible even after post-vax infection, are we going to all end up with long COVID risks? I don't think that makes any sense, Bryce. So I don't think we have any data on this. But how can this be possible, that we're all going to end up with long COVID risk? It doesn't make any sense. Just step back and think about what we know about viruses. It doesn't make sense to me at all. So I think something's wrong and we need to sort it out. Did you ever ask Ian about these claims that he knew about the outbreak on December 15th? I don't know if I asked him. Did I, Amy?
I know. He acknowledges he got a phone call and they talked about that there were a few cases of weird pneumonia in China. And he said, okay, and as I said, he gets hundreds of these phone calls a day. He doesn't have any idea. And Niche was standing right there when they were talking about it. And Niche was like, so it's another respiratory pathogen. We have no idea. You get them a ton every day. You have no idea which one is going to bloom out and which one's going to be crap. So I don't see what the point of this discussion is.
Snowfox loves your purple shift. Is that a shift? I thought it was a shirt.
Yeah, he just spelled shirt wrong. It's fine.
My brother got the alpha variant before vaccines were available and was quite ill. He now has had 3 AstraZeneca vaccines. Does he have more immunity because he had COVID before being vaccinated? Yes.
Yes.
It's really good immunity. Yes. I agree with prophylactics being a bad idea, but the geography of the early pandemic is unmistakable. I think you haven't seen how far back the timeline goes in China. Okay. All right, we're wrapping it up here. Thank you, Datebio, for your contribution. Are the Pfizer and Moderna vaccines comparable in strength? There's a huge dosing difference. Daniel has said they're close enough. It's good enough. And he doesn't think it's worth picking one or the other. All right, 1080p is not worth it. Fine, we'll stick with 720. We'll fix Amy's fuzziness other ways. Have you considered putting together a book of COVID poetry as a fundraiser? I think that would be cool. I don't know if I have enough, frankly. We do have a lot of poems, but then I have to get everyone's permission and so forth, and I just don't want to deal with that. The under 65 effectiveness of Paxlovid was from the Claylid study presented on Quiv 908. Okay, thanks. What would happen if someone had COVID and monkeypox? Well, it's a DNA and an RNA virus, so they're not going to merge. You might have different symptoms, though, as a consequence of the two infections, right?
Mm-hmm.
Is the FDA requiring you to take a fourth shot for work?
Not that I'm aware of.
Interesting.
I'll find out on Tuesday what their policy is. That's when we discuss it.
Yeah. What's the process of determining which of the millions of different viruses are attributed to symptoms? How do we know we are sequencing the right virus?
Well, that's the problem.
Oh, if you think you're sick,
no,
you
just
do a
think
PCR.
about it. Right, but just think about it. When we talked to Palanch years ago about 68 and AFM, his response was, in those samples, there's a ton of crap, meaning that there was a ton of other viruses and shit in there.
Mm-hmm.
Right? You really have to have a lot of numbers in order to really be, and it's not millions of viruses, it's patients.
Oh, what are we talking
that have
about?
the same symptoms.
I mean, any one person has many different viruses in them, but we don't deep sequence everyone. If you show up with symptoms, respiratory symptoms, you can get, if there's not a COVID pandemic, you get a rapid flu test and you see if you're positive for that. And if you are, then you get antivirals. That's how it works. You take, and if you have a respiratory disease, you can do a respiratory biofire panel and figure out which of those viruses you might have.
If you said, I have this respiratory disease and then I have neurological complications that look like polio, they went and deep sequenced all the kids. And they found that there was a ton of crap in there. Talk to Palanch. Remember that we were at a meeting with him.
I understand, but these are for... Yeah, but I'm talking about the common viruses that cause...
Yeah, but that's not... I don't think that that's what they're talking about. I think that they're talking about when it's an unknown. When
well, let's say the first cases in China with unusual respiratory illness, right?
Yeah, but then they were able to physically identify the virus, right? They took spectrum.
Wait. So what they did was they ruled out a ton of respiratory viruses by PCR, right? All the suspects they ruled out. And so they said, so what's here? You deep sequence and you have a high signal of RNA from this virus, which is reproducing very high.
No, but but before that, they did EM on the bowel of those people and they saw the particles.
Okay, fine. Coronavirus-like particles, right? But at some point you have to identify it, right?
Right, but that's assuming that by the time you come in with symptoms, the virus is still hanging out.
Yeah,
if you just have symptoms and like for 68, by the time you developed AFM, the virus is no longer present. So you're just down to sequence.
I understand. Thank you, Margot, for your contribution. Really appreciate it. Elizabeth said, Amy, in the corporate world, non-mandatory meetings mean they aren't going to pay you, but you still have to go. I'm not sure what it means in government. Thank you for that.
I don't know. I learned some stuff. I'm going to hear the same stuff on Tuesday. It's all fine.
I remember a Jane Goodall documentary where polio went through a wild chimp population. Well, I don't think that was polio. There was no proof it was polio. She gave them the vaccine, but there was no indication that it was polio. So I'm still not convinced that there's polio in any wild animal. You know what I'm talking about, Amy?
I do, but it's just like I'm not convinced that chimps are a natural host of rhinovirus, but if you blow hard enough on them, somehow they got infected, right? Because 8% of the chimps that Anne and those people in Wisconsin looked at died, right?
Yeah.
All right, 9:59. Is it? Are we? What are we doing here, Bob?
I'm looking to see who else has donated, Amy, and then I will thank them, and then we will say goodnight.
Okay.
Okay. And here we
Okay.
have Ray Med. Thank you. Hep A and B vaccines
that's very
recommended
nice.
for travel to Latin America. No, not to Latin America, as far as I know. No.
I think Hep A is because of all the fish.
Latin America, I wasn't required to get a Hep A. South of Brazil, I was. All right, so that's 10 o'clock, and it looks like we have got all the questions answered. I want to thank the moderators for tonight. Les, Vanity Nutrition, Steph, and Tom for doing a great job. They are our immune system. They never answered what they wanted to be. Too bad. Maybe next time.
They'll get to it.
Maybe or maybe I skipped over it. Anyway, thank you, mods. Thanks, everyone, for coming and bringing your lovely community and your great questions. And we're always happy to be here. We'll be here next week. Meanwhile, stay safe, and thanks for coming. Good night, everybody.
Good night.